In this virtual lab, I'm going to walk you through the process of how we're going to isolate the DNA of this raw wheat germ. We're going to use common household chemicals such as dish soap, meat tenderizer, baking powder, which this is the sciencey kind, but you probably have some baking powder at your house already. And the last thing we're going to use is rubbing alcohol, or isopropyl alcohol if you want to sound like a nerd. This wheat germ is the baby embryo inside of the seed of that wheat kernel. And for the purposes of this experiment, it's really a good source of DNA because wheat, the kind that we are going to use, has 8 copies of each chromosome. It's important that this is the raw wheat germ because when they toast the wheat germ, that heat will actually denature and break down the DNA. So you got to make sure and get the raw. So we need to get our wheat germ together. And because I'm in the lab, I'm going to weigh it out using my balance here. But if you're following along at home, you can just use your regular like measuring spoons and measuring cups and stuff for this. So first thing I'm going to do is I'm going to weigh out about 2 grams of my wheat germ. So I've got about 2 grams of my wheat germ. That looks like about 1.5 tablespoons meat tenderizer. Okay, this has enzymes that's going to break down proteins that are going to get in the way of my extraction. I got 2 grams of my meat tenderizer. This is a lot denser, so this is probably about 1/2 teaspoon. So if you're following along at home, 1/2 teaspoon of meat tenderizer is what you're going to need. The first thing that we need to do is get the DNA out of the cells of the wheat. We're going to do this by doing cell lysis, which is just fancy science talk for we're going to break the cell down. I'm going to use a beaker because I'm in the lab. If you're following along at home, you can use a measuring cup or a little glass or whatever you have. Okay. I'm going to add about 45 milliliters of water. If you're following along at home, that's about 1/4 cup of just regular tap water. Okay. Boom. Next thing I'm going to do is take my wheat germ and I'm just going to sprinkle that just across the top of my surface of my water here. Okay. Get it something like that. Dawn or Palmolive work the best for this. Now you're like, cool, I'm doing this at home. I don't know what 3 milliliters is. It's going to be a heavy 1/2 teaspoon or so. I'm using a disposable pipette for this. They've got these marks on there that allow me to measure how much I'm getting. So there's my solution. I'm going to give that a little mix. Just stir this up. You can use whatever tools you have around your home. We're going to keep this in a hot water bath for about 10 minutes. We want the water to be at a temperature between 50 and 55 degrees Celsius. For us Americans, that's about 120 to 130 degrees Fahrenheit. So see you in about 10 minutes. Like the phospholipids in the cell membrane and in the nuclear envelope, the dish soap that we added is amphiphytic, meaning that portions of the molecule are hydrophilic and portions are hydrophobic. And so that detergent is going to help break down those membranes, thus releasing our DNA. The heat that's applied is used to assist in the softening of the cell membranes, enhance the activity of the detergent, and denature enzymes that might otherwise damage the DNA. Now we're ready to add that 2 grams or about 1/4 teaspoon of our meat tenderizer. We're just going to sprinkle that on top there. We're going to add that. Give it a stir. And we're going to put it back in the water bath for about 10 minutes. The sodium bicarbonate solution that we added to our extraction acts as a pH buffer to keep that pH at a happy level around 7. That's the pH where the DNA is the most stable. And the enzyme that's present in our meat tenderizer is most effective at a pH of about 7. So the meat tenderizer contains an enzyme called peptin, which is naturally present in papaya, pineapple, and other fruits. The optimal conditions for the peptin enzyme is between 50 and 65 degrees Celsius and a pH range of about 5 to 8. The peptin completes the breakdown of the nuclear envelope and removes some of the proteins that coils up the DNA. At which point then, our DNA is free and in solution. So our extraction's been sitting in the hot water bath for about 20 minutes total now. We're going to take it out and cool it down to about 25 degrees Celsius or about 77 degrees Fahrenheit. Into the ice. The final step requires cold alcohol. DNA is hydrophilic in nature due to that negative charge phosphate group. This is going to allow DNA to interact with a partial positive area of water, allowing it to dissolve by forming those weak hydrogen bonds. Isopropyl alcohol has one polar bond and many non-polar bonds. When adding the water and the DNA, the alcohol will disrupt the hydrogen bonds between water and DNA, causing it to interact with the alcohol instead. This causes the DNA to not be water soluble, and it will precipitate or become visible. So now here's our extraction at this point. We're going to add our alcohol now. You're going to add about a tablespoon of your ice cold isopropyl alcohol. I've already got mine here. Oops, still got some ice stuck to it. So it's ready to go. At this point, we want to be really careful not to mix or twirl our solution. We want to keep those layers. And I can already see the DNA coming out of solution. So here we have a layer of all of the wheat germ. Then we have this watery solution. Up on the top is our alcohol, and all of those stringy things that you're seeing are the DNA. So the snotty white, stringy white stuff that we're seeing, that is the DNA from our wheat. All right, so for the last thing that you're going to do is for your results, you're going to take a photo of your extraction if you followed along. And if you didn't, you can draw this and label each of those layers.